Aflatoxins in food and feed. Hello, everybody. Today we are talking about aflatoxin. Aflatoxin is a type of natural toxins produced by fungi found in crops such as corn and cereals. Why is it important? Aflatoxin is primarily found in agricultural products, such as crops, which comprise the majority of the world's diets. That is, we somehow posed a risk of aflatoxin-related health problems. Aflatoxin also imposes a significant economic cost, destroying an estimated 25% or more of the world's food crops each year. Before we learn more about its consequences on consumption of contaminated food, let's first familiar with the toxin. What is aflatoxin? Aflatoxins are mycotoxins produced by certain fungi that are capable of causing serious illness in animals and humans. Among the various types of mycotoxins, aflatoxins are extremely toxic and have been found to contaminate a wide variety of foods and feeds, including peanuts, nuts, figs, corn, rice, spices, dried fruits, meat, and dairy products. Two closely related fungi are primarily responsible for the production of aflatoxins with public health significance: Aspergillus flavus and Aspergillus parasiticus. AFB1, AFB2, AFG1, and AFG2 are four major aflatoxins of the identified 20 types that are frequently found contaminated in agricultural commodities worldwide, particularly in tropical and subtropical regions. Additionally, aflatoxin M1, AFM1, a product of aflatoxin B1, AFB1, metabolism, has been detected in milk from areas with a high level of aflatoxin exposure. Aflatoxin M1, AFM1, is a toxin produced by mammals following their consumption of AFB1 contaminated feed or food. Humans may then be exposed to this aflatoxin through milk and milk products, including breast milk, particularly in areas where substandard grain is used for animal feed. The extent of fungal growth and aflatoxins production in cereals depends on temperature, moisture, soil type, and storage conditions. Aflatoxin can be produced in warm, humid storage environments and in tropical and subtropical geographic areas. Moreover, global climate change is one factor that promotes growth of molds and production of the toxins. It alters the temperature and water activity in the environment, which further influences the gene expression to produce aflatoxins. The safe level of aflatoxins for human consumption lies in the range of 4 to 30 micrograms per kilogram. Acute exposure to high doses of aflatoxins can cause aflatoxicosis, with the liver as the target organ, resulting in severe liver damage. Aflatoxins impair the liver's normal functions, including carbohydrate and lipid metabolism, as well as protein synthesis. Chronic or prolonged exposures result in cancer, impaired protein synthesis, impaired blood coagulation, toxic hepatitis, and possibly immunosuppression. AFB1 is known to be carcinogenic in humans. Exposure of the toxins has been linked to an increased risk of liver cancer. Adults have a higher tolerance for acute exposure than children do. It also causes immunosuppression, therefore may decrease resistance to infectious agents, such as HIV and tuberculosis. In chickens, aflatoxin-induced liver damage, decreased productivity and reproductive efficiency, decreased egg production, inferior eggshell quality, inferior carcass quality, and increased susceptibility to disease. Pigs are also highly susceptible to aflatoxins, with the majority of the chronic effects manifesting as liver damage. In cattle, the primary symptoms are decreased weight gain and liver and kidney damage, as well as decreased milk production. Cows are able to metabolize, process, aflatoxin. The substance, metabolite, produced by the cow after it processes the aflatoxin may then appear in the cow's milk, but is less toxic than the aflatoxin itself. Aflatoxins can be managed at every stage of production, from the field to the table. We can reduce the risk of aflatoxin exposure in two stages: pre- and post-harvest. Pre-harvesting. Plant breeding, known as genetic engineering, is the most long-term stable method of preventing pre-harvest aflatoxin contamination. The aim is to strengthen the crop's resistance to fungal infection, and/or to prevent the invading fungus from producing aflatoxins. Biological control with non-toxigenic Aspergillus flavus isolates is one strategy that has garnered considerable attention for aflatoxins reduction prior to harvest. Controlling aflatoxins through the use of non-toxigenic strains results in strains that are capable of competing with and displacing the toxic strains. Post-harvesting. Farmers or producers should pay attention on post-harvesting and storage processes. Food, especially agricultural products, should be kept in proper storage conditions, include moisture, temperature, mechanical or insect damage, and aeration. Additionally, chemical decontamination, or the use of enterosorbents, can aid in the removal of aflatoxins from foods that are already contaminated. Aflatoxins cannot be destroyed by heating or cooking. Molds do not grow only on the surface of food, they penetrate deeply into it. Consumers should inspect whole grains and nuts carefully for signs of mold, and discard those that appear moldy, discolored, or shriveled, as well as diversify the diet to reduce the risk of excessive aflatoxins exposure. Lastly, increasing public awareness for implementing GAPs, GMPs, and GSPs is critical for preventing aflatoxins contamination in food and feed.